what we covered in lecture this morning. And then I'm going to go
Yes.
ahead and go over with
Okay,
you
yeah.
the
Gotcha.
thalamus and hypothalamus
Yeah,
as
no
well
problem.
as brainstem. Although we haven't lectured on that yet. We'll do that on when we get back. So I won't see you guys for 2 weeks. Because next week is spring break, and then the following Monday is Cesar Chavez Day, which, so we're missing one of our days, and then we have class on Wednesday. We still end up having 5 days for this unit. Typically, every 5 days we have a test. So we were scheduled to miss a day. There's just no way I could have covered this in 4 days, even, and it's not material I want to tell you to study on your own. But what that means is we have our last lecture lab day on a Monday, and then no weekend in between, and our exam on Wednesday. Not the week we come back, but the following week. So you need to plan ahead. Take spring break, you guys need it. But that Cesar Chavez Day, if you can, I would turn that into a study day. All right, so look ahead and see your schedule, because you're not going to have a weekend between learning the eye and the ear and endocrine system, and then we have our test 2 days later. So plan ahead for that.
Wait, but our test is on Wednesday.
Our test is on a Wednesday. So we come back on a Wednesday. That day we'll do brainstem and cranial nerve. Then we have a weekend. And then that Monday we'll do eye, ear, and endocrine. And then that 2 days later we have our test. Okay, so you need to so that's why I'm kind of moving ahead a little bit and going over brainstem with you today, even though we won't have covered it in lecture. So when we come back, the day we come back, we do brainstem, cranial nerves, and a brain dissection in lab. So all of that is why I'm going to go ahead and do brainstem with you today, even though I haven't lectured on it. So it gives us a little bit more time in lab. We can kind of move maybe part of the special senses up a little bit, so that last day you don't feel so crammed without some time for that to settle in. So look ahead at your schedule. All right, if you can study during spring break, that would be great. I was a student who loaded up my suitcases and flew home with all my books and never opened them. But I did feel guilty at least. But at least that Monday, all right, plan on that being a study day. Our next quiz, because we have the brain dissection, I don't have a quiz scheduled for the Wednesday that we get back, because our day is going to be full. So our next quiz is going to be the last day before the exam, and it's going to cover at least one ventricle structure question from Monday. It'll cover brain, brainstem, and cranial nerves, and it's designated that way on your schedule. Okay, so again, kind of keep up with that material. So we're on page 118, 119, and we'll do page 120 and 121 as well. There's not much to page 119, okay? There really isn't, so I think you'll find that too difficult to follow. So the first structure is just looking at a gyrus. Remember I said a gyrus is just the fold of gray matter. The sulci are the two valleys between that fold. We have a couple of gyri that you need to learn the names for, but just as a general term, that's what a gyrus and sulcus is. If you look at this cut surface of the brain, this would be a gyrus, and the groove on each side would be the sulcus. Right? For those specific components, on your diagram, F and G are the pre- and post-central gyri, and H and E are the sulci that we're going to learn. So now when you look at the diagram on the lower left, is showing the brain on an inferior view like this. So you have to flip it over to think about it. So when you're looking at it like this is going to be the right hemisphere, and this is the left, if you think of it the way it's going to fit in the brain. So L is showing the right hemisphere, and M is showing the left hemisphere. Remember the cerebral hemispheres from our embryonic development come from the telencephalon. So if we're matching up lobes, frontal lobe, it says that on your lab manual, so the word telencephalon is right there in parentheses by cerebrum. So you would be matching frontal lobe, precentral gyrus, temporal lobe, lateral ventricle, all of that are components of the telencephalon. Now, if we separate the cerebral hemispheres, all right, in this fashion like that on the sheep brain, when we do our sheep brain dissection, you'll be able to do that, but not completely without cutting it, because the two cerebral hemispheres are going to be joined by the corpus callosum. But when we do separate them, that space is identified as the longitudinal fissure. That's letter I on your diagram. So I can mark that either by indicating right here, identify this space, okay, between the two would be the longitudinal fissure. Or I might slip a piece of paper into this gap, like that, and say identify that space, and that would be longitudinal fissure, okay? Then the sulci are available on the lateral view. So the lateral sulcus, you don't have to include the word cerebral, but I would mark it wrong if you did. Lateral sulcus is fine. And that is this one right here. So that's going to separate temporal lobe from frontal and part of parietal, and that is letter E. Now, the diagram on the right with letter E shows the cerebellum, so it makes it easier to figure out that that's the posterior aspect and this is the frontal aspect. The diagram on the left doesn't show the cerebellum. So in figuring out which is frontal lobe and which is occipital lobe, you need to look at that cerebral, that lateral sulcus. And that will help you orient yourself to which is front and which is posterior, anterior and posterior in the brain. The central sulcus is passing between, separates frontal lobe from parietal lobe. And on some of the brains, that has been indicated by highlighting with pen and pencil. Okay? On other brains, not so much. So on this one, it really isn't showing. So I would not take this brain and point to this sulcus and say or point to this gyrus and say identify this gyrus. Because then you're left trying to decide if this is the central sulcus, or if that's the central sulcus, or that's, so which side of the sulcus is that gyrus? So on something like this, I would point to the sulcus as well. And say identify this sulcus, and then the other question would be to identify the gyrus, and that way you're oriented. Because the only sulcus that's there is the central sulcus. And if I asked the gyrus in front of it, it would be the precentral gyrus, and the gyrus behind it would be the postcentral gyrus. Okay? If it's been marked like this one has, okay, where the sulcus is plainly marked, then I might just say identify the gyrus. Or on the chart over here, where the two gyri are shown in different colors. So the yellow gyrus would be the precentral gyrus, and the bluish gray one would be the postcentral. So I could mark those without asking for the sulcus. Okay? But if the sulcus isn't clear, I will not ask for the gyrus without also tagging the sulcus. So on your diagram, the central sulcus is H, precentral gyrus is F, and postcentral gyrus is G. Precentral gyrus is motor or sensory? Motor. Good, because it's in the frontal lobe. And that's what's formed of our upper motor neurons. Postcentral gyrus is sensory, because it's in our parietal lobe, and that's where we process pain, touch, temperature, sensations. All right, if you would add another sulcus, in lecture I talked about the parietal occipital sulcus, and so if you want to draw it in on this lower diagram right here, you would highlight this a line right across here, and that separates parietal lobe from occipital lobe. So kind of the last third. On the hard plastic models is pretty clear. It's right here. So this right here would be the occipital lobe, and that would be the parietal occipital sulcus. So it's spelled parietal with an O, P-A-R-I-E-T-O, occipital. O-C-C-I-P-I-T-A-L. All right, so right there. And that gives you on the medial surface the best separation between occipital lobe and parietal lobe. Okay? On the lateral surface, it's unclear. It's only like an inch or so across the side. So you can see it best on the medial surface. Okay? All right, the corpus callosum. Anybody remember what type of white fibers the corpus callosum is? Association, commissural, or projection? Commissural, good. And this is corpus callosum right here. So these are fibers that are traveling between the hemispheres. It looks like a structure that goes this direction, but they're actually fibers that are going back and forth like this. So if I were to take back to this sheet, so the corpus callosum is going to be, oh no, this is somebody's quiz from the next lab. So those would be white fibers connecting the two halves, and they form the corpus callosum like that. And if you see it on the side view, the way we see it in the brain, it looks like that. Okay? But the fibers are actually going left to right, right to left. So corpus callosum is letter N. N is in nevermore. If we look, I'm going to come back to olfactory bulb. So if you look at the lobes now, and they seem to be pretty basic, but you'll be surprised somebody always misses it because they're not used to the surface I look at. So we'll look at all the surface of the lobes. So the frontal lobe is not just this front part here, back to the central sulcus. It's also what you see here. So on the inferior aspect is frontal lobe. And then if we look at the medial aspect, here's cerebellum, so that tells you it's posterior. Frontal lobe is also here. Okay? So basically all of this that I'm grasping is going to be frontal lobe. You should recognize it from any of those views. And that would be letter V on the diagram in the upper left. Okay, temporal lobe is inferior to the lateral sulcus. Its most posterior definition, there's not a distinct sulcus that separates it, but all of this, and then on the inferior view, this right here, the bump, this inferior bump here is temporal lobe as well. So essentially what I'm holding here would be temporal lobe. Hippocampus is going to be inside that. And while you don't have to identify hippocampus, we'll see it in the sheet brain. It's really cool. So that would be letter D, as in determined, in the upper left portion. Parietal lobe is going to be posterior to the central sulcus, back to about here. All right. So this portion will be parietal lobe. And when we look at the medial surface, we don't see the temporal lobe on the medial surface. It's too lateral. But parietal lobe is going to be basically where my fingers are here. So that's parietal lobe. Okay. Frontal lobe here, parietal lobe here, and occipital lobe here. You're not going to see temporal lobe on this medial view. So parietal lobe would be parietal lobe would be A, and then C would be the most posterior aspect. Again, the parietal occipital fissure, and essentially what I'm grasping here. So from the inferior surface, it would include this part back here. This would all be temporal, but back here, essentially that portion would be occipital. Okay? And when I tag it, I'm not going to go, tease you by kind of putting it right somewhere in between the two. I'll make it distinct. So C is occipital lobe. And then the insula is the shaded area, O. So on the hard brains, you can't see the insula because you can't open up the lateral sulcus. But if I open up the lateral sulcus like this, you can look inside and see that there's cortical tissue there. It's folded into gyrate. And so what you're looking at, if I pull this away and bring this out, is this structure. That's the insula. All right, so on our isolated brainstem models, that's insula right there. And that's probably how I'll tag it, because it's hard to keep that lateral sulcus open to tag it down at the base. But that would be the insula. And it's the shaded area marked O. All right, so that's what we covered in lecture today. So if you'll turn to page 120, 121, we'll look at the diencephalon. Remember that diencephalon and telencephalon developed from our early embryonic three vesicles, the most anterior being the prosencephalon. So telencephalon is going to be the lobes of the brain, the insula, the lateral ventricle. Diencephalon is thalamus and hypothalamus, which are covered up by the rapidly growing hemispheres, as well as the pineal gland and pituitary gland. Those are part of the endocrine system that we'll cover the last day before the exam. So you already should be familiar with the thalamus and hypothalamus from the lateral view that we looked at on Monday, because that space is the third ventricle. All right, so the third ventricle is the space between watered by thalamus and hypothalamus. So if I had this model on a lab exam and I wanted third ventricle and I tagged the hypothalamus, if I wanted third ventricle, I'd have to say identify this space. Otherwise I want the structure itself. Okay. So thalamus is going to be the rounded region. So it's letter C on the upper diagram on page 120. And the circle in the middle of C, letter D, is the intermediate mass. That's not formed by bridging fibers, by commissural fibers, but just two parts of the thalamic nuclei that happen to touch each other. So remember in our ventricular models, when we looked at the third ventricle, on this model, there was a hole in the middle of the third ventricle. That's actually a solid structure. It's a solid structure. The third ventricle is a space. So that hole in the middle of the model is that intermediate mass, where the two thalamic nuclei touch each other. All right? So B is the intermediate mass, C is the thalamus, and that's all you can really see of the diencephalon, thalamus, and hypothalamus in this model. We'll get to the hypothalamus in just a moment. But on this model, there's the thalamus. Go posterior, and you can see a much larger region of the thalamus posteriorly. Okay? So be sure and look at that with one of the brainstem models. Now I have a really large brainstem model here. This is the front. So all of this structure right here looks like an egg. All of that is actually thalamus. Okay? And we'll talk about the function of thalamus in a couple weeks. The hypothalamus is inferior and anterior to thalamus. So hypothalamus is letter A on your diagram. Growing out of the floor of the hypothalamus is going to be the optic nerve and the pituitary gland. So some of these models have an orange or brownish ball kind of hanging down from a wire, and that's the pituitary gland. On our sagittal head model here, you can see the pituitary gland within the cella turcica. So part of the pituitary grows out of the floor of the third ventricle from the hypothalamus. Part of the endocrine system, and we'll come back to that again the last day of lecture. Part of the optic nerve comes back and forms a crisscross. That's cranial nerves. Again, we'll talk about that when we get back. But on this diagram up here, so when you look at the ventral surface of the brain, you will see the two optic nerves coming back from the eyeball towards the center, and part of the nerve fibers will cross and go to the other side of the brain. Part of them do not. And they do this pattern. So this is optic nerve. We'll talk about it when we get back because it's part of the cranial nerves. Where some of the fibers cross is identified as the optic chiasma. And it's seen as letter F on the upper diagram where it's been cut and you're looking at a sagittal view of that cut surface. Okay? You cannot see it on the lower diagram because it's covered up. You can see the X, but the optic chiasm itself is covered up by the stalk of the pituitary gland. And then O would be the optic tract. Now these are sensory fibers, and I already talked about the thalamus being a sensory relay. So all of our sensory information except for smell, those neurons go to the thalamus and synapse with other neurons that if it's hearing will go to the temporal lobe. If it's smell or pain or touch or temperature, not smell, vision will go to the occipital lobe. Pain, touch, and temperature, parietal lobe. All right? So if you follow on these brainstem models, it's a little harder on the whole brains, but on the brainstem models, if you follow that optic tract back, it'll take you to the posterior thalamus. It'll take you to this large fat part of the thalamus. So optic tract, you can just follow it back and around and end up in the thalamus. It helps you find that. Okay? Again, you can't do it on the solid half brains. You just can't get to that point. Alrighty? So O is optic tract. Mammillary bodies are part of the hypothalamus. Remember I talked about hippocampus and the temporal lobe? And when you lay down short-term memory, there are nerve connections between the hippocampus and the mammillary bodies. Okay? Mammillary bodies are also associated with a part of the brain known as the emotional brain. Interestingly enough, if they're damaged, people show abnormal behavior, a lot of licking. So it's part of, we don't really talk about much of those structures, but that part of the mammillary body. So from the ventral view, you can see them in cross-section. It's not labeled in cross-section, but in the ventral view, they're numbered letter N. And so they're found just between the optic tracts. Somebody thought they looked like small breasts. And so they're called mammillary bodies. All right? And I'll show them to you on this large brainstem model. On some of the smaller brainstem models, they're also easily visible. The other, again, the large brains, they're kind of compressed and that space is tight to see that area. So now let's move into the midbrain. The brainstem is divided into midbrain, pons, and medulla. So again, we see our motor structures are primarily ventral, and sensory structures are primarily dorsal. So from the front, we can see, my way here. Remember I talked about the motor pathways being two neurons, the cell bodies. If I decide I'm going to extend my fingers, cell bodies in the precentral gyrus fire, and that sends an upper motor neuron down through my brainstem, it crosses over and goes down my spinal cord, and synapses with lower motor neuron cell bodies in my anterior gray horn. So all those fibers descending down form a bundle of upper motor neuron fibers coming down to the front of the brain. And that forms a structure forming a V on the ventral surface. And this is known as a cerebral peduncle. It's part of the midbrain. And it's a bundle of projection fibers. These are fibers that are projecting from the brain to the spinal cord. And they're just a bundle of upper motor neuron fibers. So let me take this large brain model. Again, this is ventral surface. All right. So all of this is cerebral peduncle. Those are upper motor neurons. And right there are the two mammillary bodies. So those are going to be found in the V created by the cerebral peduncles. So my fingers are on the cerebral peduncles pointing to the mammillary bodies. Okay, again, ventral. Cerebral peduncles are motor. Okay, right there. Did you see it? Okay, and then my fingers are pointing to the mammillary bodies. So these are the peduncles. Those two bodies in the middle are the mammillary bodies. Okay? So mammary bodies, and I'm pinching the cerebral peduncles. Okay, and mammary bodies are those two bumps in the center. So pinching the cerebral peduncles, those two bumps in the middle are the mammary bodies. Right here. So, pinching the peduncles, and those two bodies right here are the mammary bodies.
So these are the peduncles there?
These are the peduncles right here. Right. Okay? Those are upper motor neurons. Okay?
Yeah. Those are
All right, now, notice that just inferior, so in your diagrams, the cerebral peduncles are G. I'm not going to tag them on a brain model like this. It's just too hard to see down in there, all right? So I'll use a brainstem model like this or one like this. They're a little bit easier to see. Now, so inferior to that, we have this large rounded, I call it a pregnant belly or a beer belly. The reason it's large is because not all of these fibers pass all the way down to the spinal cord. Some of them will stop and synapse with fibers going to the cerebellum. So the presence of these cell bodies creates the large swelling of the pons. It's actually that surface is the basal pons, but it's the ventral surface because it's motor neurons. All right. So we see this area here, ventral, which is basal pons. If you turn it over, the area behind it is the open fourth ventricle. The roof over that would be the cerebellum. Okay? So there is no dorsal structure for the pons. Which reminds me, I skipped the dorsal structures for the midbrain, so let me go back to that one. If you look at the posterior surface of the brainstem, you'll see two swellings. Now, if you're looking at a sagittal view like this, find the cerebral aqueduct that we studied on Monday, and you'll see two little bumps above it. So here's the cerebral aqueduct, and here's one bump and another bump. Okay? If we put the two halves of the brainstem together, we actually have four. Two on the top and two on the bottom. This is a posterior view, or a dorsal view. And so those, here's my other half of the brainstem. Is there a brainstem back there that's two parts put together? Because we have a complete brainstem and then two halves that.
So it's going to be
There we go. Okay.
superior.
Yes. So now, this is a posterior view, fourth ventricle, superior, inferior colliculi. Okay, and you will see that on the sheet brain as well. So, superior colliculi, inferior colliculi, fourth ventricle. Did you see it, Todd?
I didn't.
Okay. So, superior colliculi, inferior colliculi, fourth ventricle. Fourth ventricle is shared by pons and medulla. Superior and inferior colliculi belong to the midbrain. Superior colliculi, inferior colliculi, the diamond-shaped opening is the fourth ventricle. That's the only posterior structures you need to identify for the brainstem. Okay? So again, superior colliculus, this would be thalamus right here, like I drew on the board. Superior colliculus, inferior colliculus, fourth ventricle. So you'd have to turn this over like this. Superior colliculus, inferior colliculus, fourth ventricle. Okay? Superior colliculus, inferior colliculus, fourth ventricle. Okay? Now, I haven't given you much function, but I just need to warn you, on spring break, I'm not sure where you're going, we're headed for San Diego, so it's not supposed to be warm, but let's say it's warm and I'm out there on the beach. Haven't seen my husband in a couple of weeks. So we're laying out there, you know, reading our summer novels, and this guy goes walking by. Now, my husband's pretty good shape, okay, for being 60. But let's say that somebody in better shape goes by, and I can't help it, I just start to follow. I can blame it as a reflex on my superior colliculus. Because the superior colliculus is hooked up with that optic tract. And when something crosses my field of vision, there's a reflexive movement that my eyeballs and or my head turns to follow. Okay, so if you're out to dinner and the same thing happens, blame it on your superior colliculus. The inferior colliculus is involved in a reflex where you turn your head for sound. So you hear a sound and you just automatically turn to hear better. Okay, so those are sensory reflexes, superior, inferior colliculi, dorsal part of the midbrain. So superior colliculus is shown in the sagittal view as letter D, and inferior colliculus as letter E. All right, let's skip the cerebellum. We just did the pons. We'll do the medulla, and then we'll go back to cerebellum. So remember these motor fibers? The upper motor neurons formed our cerebral peduncles. They pass through the pons, synapsing with cell bodies that go to the cerebellum that make the swelling of our pons. But then they keep passing inferior, all right? So here's the bottom of the pons, and those motor neurons keep descending down. There's fewer of them because some of them stopped in the pons. And so these create, in the medulla, a longitudinal swelling known as the medullary pyramid. And that would be letter I in your diagram. Now I will lecture on this on Wednesday when we get back, all right? Because the afternoon class, I guess, I'm not sure how much of that he'll go over. But I wanted you to be familiar at least with this structure, so we don't have a ton of material to go over new. So just below the pons, on the ventral surface, because these are still motor neurons, we have the medullary pyramids. On our big brainstem model, it's these two struck columns right here. Okay? Just continued by the same fibers that form the cerebral peduncles, pass through the pons, and now form the medullary pyramids just below the pons. Letter I. Now Dr. Carter has also put in the lab manual the olive, which is letter J. We're not going to talk about the function, but she put it in there to help you find the hypoglossal nerve, the motor nerve for the tongue muscles. So just lateral to the pyramid is a small oval swelling that looks like a large olive, or small olive, depending on the size of your model. That's letter J, and the hypoglossal nerve we'll talk about and when we get back is the nerve fibers between those two. All right. So those 2 pages, okay? And then we'll do the lecture concepts when we get back. Oh, cerebellum, thank you. I forget to do that. So here's the cerebellum. We'll talk about its function when we get back. It's primarily for balance. These would be the cerebellar hemispheres, the lateral large aspects. Cerebellar hemisphere, that doesn't show both, but L would be the left cerebellar hemisphere. There are 2. They fit together like this. Okay? And then between the 2, but that way, they fit together like that. In between the 2, more shallow, is the vermis. So the vermis is letter M. All right, so it's like we have a So here would be a cerebellar hemisphere, here would be another cerebellar hemisphere, and the vermis is this middle region. All right, it's been cut in the cross section. So that's letter M. Now when we cut through the vermis, you can see a pattern of white matter that reminded somebody of the branches of a tree or maybe the root system. And so it's called the arborvitae, which means living tree. All right, so that's the white matter pattern shown as letter K. Now I'm done talking. Unless I'm answering questions. But those are the 2 pages that we're covering today. And then I will lecture on them, and we'll go over them again when we get back.